All right, we are into our last chapter. I know everybody's real sad. This is the last one. This chapter is going to talk a lot about epidemiology, which is the study of like tracking disease and outbreaks and ways to describe that. So we'll talk about several examples here, and we'll talk not just about what it is, but why it's important. And then we will finish up with a little material about COVID-19. There are some important pieces in there, but then I will go into extra detail that I probably won't test on, but just if you're interested in that, we will have that out there. I know as we get further from the pandemic, people want to hear less and less about it, but it is still important to understand what is going on with it. Okay, so we're going to start by talking about the basics of epidemiology. Talking about things like outbreaks and endemic diseases, epidemics, pandemics. What are those? Describing those terms. Talking about how we track what we call healthcare associated outbreaks of disease, also disease in the community as well. And then we'll talk about some basic ways that we track this. We'll talk about some factors that influence outbreaks of disease, particularly related to technology and as the earth changes. And then we'll talk a little bit about what influences new and emerging diseases. Okay, so here's a case study. We have, it's 2015, we have a woman in Brazil. She's in the 4th month of her pregnancy, and she wakes up with a fever, fiery red rash, pain in her joints. So that is kind of general, right? We've seen a lot of diseases that might have this, aches and pains, things like that. So clearly this is the immune system responding to some sort of infection. The symptoms go away within a couple of days, so she doesn't think about it anymore. 5 months later, when she delivers her baby, everyone knows that something is wrong with her child. Her son has a very small head, and because of this, there will be incomplete brain development, which is something we call microcephaly. So she's, of course, devastated by this, but what caused this? Well, we're going to talk more broadly. Here's an image of microcephaly, so you can tell the proportions here are quite off for this child. The head is exceptionally small. In 2015, we have Dr. Vanessa Vanderlind. She's a neurologist in Brazil, and she notices that there's this increasing number of newborns with microcephaly. Okay, in and of itself, that is an observation, but we need to look at data, right? Is this just a random fluctuation, or is this some actual cause of this? Normally, she would see less than one case a month, so it would be one case every few months. Now she's got a spike in it. That doesn't mean that there's some related cause here. There are statistical fluctuations, but as the months go on, we'll continue to see these numbers staying high. So they're trying to figure out what is the cause of this. So they do serological tests, right? We look for things that would normally cause this: measles, toxoplasma, things like this, but they don't find any causal agent, but they do see a pattern of what we call calcification, which is literally basically the brain is turning to bone, kind of. It's got calcium deposits in it. This often happens in a condition called viral necrosis. So viruses can cause this. So we suspect a virus. Vanessa, her mother, Anna, is a neuropediatrician in another hospital, and they talk on this. And Anna says, "Oh, I've also seen many cases of microcephaly. In one day, she saw 7 cases." So now we have 2 separate incidents of this, and we start to get a little more suspicious. Is there some sort of single cause here? And as this goes on, we have many of those mothers reporting that they did have that rash and those symptoms early on during pregnancy. So it leads us to believe that there might be an infectious agent that is involved in this, and it's probably new because we haven't picked it up on any of our standard tests. Okay, so what do we do here? Well, she's going to alert state health authorities about this spike. And that is going to start a long process where we're collecting data. And it will turn out that this gets linked back to a virus called Zika. So you may remember this Zika virus outbreak. Viruses, particularly new viruses, are difficult to identify because you have to do electron microscopy, which is not something that most facilities are equipped to do. In Zika's case, it's an RNA virus, it's mosquito transmitted, and it can be transmitted from adult to adult, and it causes that fever. But in pregnant mothers, it can infect fetal brain tissue and lead to this development of microcephaly. So in the outbreak between 2015 and 2018, at least 4,000 babies in Brazil and 116 in the US were born with microcephaly. These US cases were usually people who traveled from Brazil to the United States and then gave birth here. But because the southern hemisphere of the Americas and the northern hemisphere of the Americas is linked, we worried about this being brought from Brazil up through Central America into North America. So in all the data that was collected, it was estimated that about 1 in 7 babies that were exposed to Zika virus in the womb developed at least one developmental defect within a year. So quite severe. Fortunately, this has dropped a lot because we recognized that it was a virus that was causing this, and we recognized that the virus was being transmitted by mosquitoes. So we implemented mosquito control measures and personal protection strategies, like using insect repellents and things like this. So all of this relies on collecting data, noticing patterns, and then tracking them back to the source. In this case, the World Health Organization and the US CDC were integral in monitoring this to basically figure out where is it. They would collect mosquitoes and screen them for the virus and see how far has it spread. Do we need to do any sort of controls or quarantines or things like this? When we break it down, right, we talked about this way back at the beginning of the course with Florence Nightingale. Infectious diseases kill more people than a lot of things, right? We think about war. War is devastating. Lots of people die in war. But infectious diseases have killed more people than all of our wars combined. So trying to find links between disease and the organism that causes it and how that is transmitted is critical in stopping that transmission process. Organizations like the WHO and the CDC, they're really key in gathering this data, right? We need large-scale data to figure out these patterns. It's no good just having 1 or 2 individual observations. We want to look broadly and analyze large amounts of data. That relies both on the big organizations to gather and collect that data and organize it and interpret it, but also individual small institutions and really the people collecting samples and stuff to report that stuff back to the big institutions. So in 26.1, we're going to talk a lot about the basics here of epidemiology. No surprise, there's going to be a lot of data collection required in epidemiology, but that data can be taken and experiments can be performed with hypotheses that are developed using that data. So we'll talk about the chain of infection. We'll talk about 2 types of epidemiology, descriptive and analytical. There are different branches of it. Then we'll talk about some terms that you probably heard, but we need to make sure that we have them properly defined. Endemics, epidemic, disease, pandemics, things like this. And then we'll talk again about prevalence and incidence. And then we'll talk about how we do surveillance for disease and microbes and things like this. So epidemiology, if we want to get right down to it, right, literally the name is the study of that which befalls man. That's a very prosaic way of saying it, right? It's studying disease outbreaks, incidence, distribution, and then trying to figure out possible controls for disease and other factors in health. So We're at the core tracking disease, right? So where is it coming from? Where will it spread to? Trying to project into the future. And how can we possibly stop that? Are all questions that epidemiology tries to address. One key thing here is understanding what we call the chain of infection. And this is the relationship between several factors. We have the organism that causes a disease. That's a microbe of some sort in our cases. There might be virulence factors in there. How invasive is it? Ability to be transmitted. Then we have reservoirs, places that it normally hangs out. These could be in animals, humans, in the environment. We've talked about this previously. Then the next link in the chain is transmission. So moving from individual to individual, so reservoir to a new host, or person to person, through inanimate objects, through the air. Is it vector-borne? This is an important part of the chain of infection. And then finally, we have the susceptibility of the host. How is it getting in? Do you have a cut on your arm? Are you immunocompromised? Are you young or old? Things like this. Each of these links in the chain is tied to the others, and all of these factors influence how a disease spreads throughout a population. Now, obviously, somebody had to develop this science, and oftentimes John Snow, not that John Snow, John Snow from England in the 1800s, he's often referred to as the father of epidemiology because he was really key in tracking disease spread. We also had Florence Nightingale, who was very instrumental in collecting data and displaying data, particularly to influence kind of epidemiology as well. John Snow was interested in an outbreak of cholera in London in 1854, and he believed that it was waterborne and possibly being spread through the water system. So he made a map, and he mapped all the cases with these little tick marks here, and he noticed that the cases tended to aggregate around one well in the city. So other wells were distributed throughout the city, but this one well had a large number of cases surrounding it. Okay, so he's collecting data on cases, but importantly, he's collecting more data than just that cases have occurred, where they've occurred, who they've occurred in. This is all epidemiology. And then he's plotted that data on some sort of graphic display here, right? Data is no good if we can't interpret it. And to do that, graphics are key. So this map was key. And he used that to localize all of these cases around a specific well. And he argued that the well might be the source, and nobody really listened to him. Like, yeah, whatever, Snow, go on. So what he did is he went out and he took the handle off the well and noticed that the epidemic of cholera went down at that point and eventually stopped. So a drastic solution. He developed the hypothesis and he went out and tested it. And in this case, this is before the organism that causes cholera was even identified. So we didn't know about Vibrio cholera, the bacteria, until 1905, 50 years after this. So this was key in seeing a pattern of disease. We don't know the organism, but we believe we know the cause and taking steps to do that. It's not always the case that we know the cause of a disease. We're getting much better at identifying them quickly, but it often takes time. So epidemiology works at a bit of a higher level. It kind of works backwards. It says, we know this is happening. Why might this be happening? We break epidemiology down into two broad groups. We start with descriptive epidemiology. This is going out and actually collecting the data. So figuring out where the new cases are, how many of them, who they occurred in, where, when. Do all these people have similar factors? Are they of the same income bracket? Do they shop at the same store? Do they work at the same job? All of these things are critical. That might sound boring, right? Going out and just taking down names and numbers, basically, right? But it's the most important part of epidemiology, because without all this information, we can't do the second part of epidemiology, which is analytical epidemiology. Analytical epidemiology uses the data from these surveillance practices where we're going out and collecting the data, and then it takes that and formulates hypotheses about what might be causing the disease outbreak or possible risk factors involved. It goes and looks at all the information and says, hey, we noticed that people that work in this specific factory all have this specific disease, or everyone with the disease works in this factory is more likely. Or in reality, it's more like if you work in this factory, there's a 60% chance that you're at risk of this, right? 60% of the cases come from this factory. It's not always clear cut, but we use that to develop hypotheses from this. But as we know, hypotheses have to be tested. We can test them in various ways, and these get a little muddied. We're not going to worry too much about these definitions. I just want to present some possibilities here for you. Okay, so we can do experimental studies. This would be like if we have a new vaccine. We could take the vaccine and take a group of people and give 50% of the people the vaccine, and the other 50%, we could give them a placebo or not vaccinate them at all. And then look and see who gets the disease. Does the vaccine group actually get the disease less often? This would be an experimental study. As you can see, there are ethical conundrums here, right? Can we ethically not give people something that we believe will protect them from a disease? Well, if it's a deadly disease, that's a bigger risk than if it's a vaccine against the common cold. We don't have those qualms. So we will see there are other ways of testing this that don't quite do this direct one-to-one comparison here. We also have cases where we can't go out and directly test things, right? We can't put cholera in a well and test to see if that causes an outbreak, right? That just doesn't work. So the observational study is where we observe the occurrence of disease and try to control for different factors and figure out what's going on. So we have two types of observational study here. So in the cohort study, we first observe who's been exposed to a potential pathogen. And then we look at that group, that cohort, we look at who in that group developed disease, and we try and figure out why they developed disease. We also compare that against an unexposed cohort, so people that live in a completely different area. And this kind of helps control for common factors. Is there anything, that this one group of people in this area were doing that led to them getting the disease versus the other group in the same area that didn't get the disease? And then we control for that kind of by using people that are not in any way exposed to this. How are these groups the same? How are they different? The other example is what we call a case control study. Here we start with people with a disease. So we go, hey, do you have this disease, hepatitis or something like that? And then we recruit a group of people who don't have the disease. Then we look at their previous histories, where they've been, what they've been doing, who they work with, what kind of activities they engage in, and we try to identify differences between those groups that may have led to the disease. So in this case, right, a case control study for hepatitis, say it's being caused by intravenous drug infection, right? We would control for things like, do you use intravenous drugs? We would probably find people that have hepatitis are more likely to have engaged in using needles for intravenous drug use than people who don't have hepatitis. But again, it's a statistical game here. Okay, so I hope that kind of explains different ways we can go about using analytical epidemiology to test hypotheses. Now we're going to talk about some very critical terms here. Okay, so you've probably heard this now. We have a term called endemic. This is used for a disease that is native to a particular area, and we see it at a steady low level in that area. This is what we call an endemic disease. In the United States, at least in the past, chickenpox used to be an endemic disease. It occurred at a relatively stable rate in the United States. When we see an increase in the number of cases of a disease above the endemic level, we call this an outbreak. We group outbreaks kind of on their severity and how broad the area the outbreak occurs in is. So when we have what we call a local outbreak in a small area, this can be classified as an epidemic when cases rise quite rapidly and the area geographically that is affected starts to widen. So let's say, for example, for whatever reason, there were no chickenpox in Canada and there were chickenpox in the United States. If we saw that the number of cases started to go up and we started to see cases in Canada, we would call this an epidemic. This can have many causes. If the reservoir for the disease increases or the infectious agent evolves or people change how they're interacting and things like this. So there can be many different causes of epidemics. When we get to a point where an epidemic is out of control and it spreads across continents, then we classify it as a pandemic. Bubonic plague, we do have a pandemic technically right now. Madagascar, some of Africa, India can all have this. HIV/AIDS is a pandemic. Influenza can be pandemic. We see that about every 30 years. And of course, our COVID-19 SARS-CoV-2 virus caused a pandemic. So these are relativistic terms, right? And the actual numbers are going to vary per disease. For meningitis, we consider it an epidemic if we have above 15 cases per 100,000 individuals. Where we draw that line depends on historical data and kind of some arbitrary calls in these cases. So if we look at a graph here, right, an endemic disease would kind of fluctuate up and down, but if we graph the average here, it would be pretty steady state. When we see a uptick in a location and it maybe spreads slightly, we call that an epidemic. If this increase were to spread across continents, we would call that a pandemic. The CDC has a book called the Yellow Book, which they update periodically, that basically is information on endemic diseases for travelers. So if you're going to travel to another country, you can look at the Yellow Book and identify what diseases are endemic to that area, and then you can take precautions, right? So if you go and read that you're going to somewhere in Africa where malaria is common, you could start taking anti-malarials or make sure you have a mosquito net or things like this. So we collect this information, the CDC, not just because it's interesting, but because it can be useful for people because we have global travel. When we're tracking diseases, generally we try to do things to mitigate the outbreak or the spread or things like this. So we need to track certain statistics, and we've talked about these previously. We have measures of prevalence, which is the total number of active cases in a population. Then we have incidence, which refers to the number of new cases of a disease in that population for a specific time frame. Remember that these terms get normalized per 100,000 individuals in the population. So we can compare small towns versus big cities and things like that. So it's not just raw numbers, it's per 100,000 individuals. In these cases, the time frame that we're looking at for incidence, we might look at per year or per month or per week. It depends on the disease and what we're trying to assess here. So let's look at an example here. We have Happy Valley Rest Home. Sounds nice, right? Unfortunately, we have 10 new cases of pneumococcal pneumonia that occurred in March. For the entire month of March, the total number of active cases was 23. So some people had a case that stretched over from the previous month. So they had 200 residents that month. So in this case, our incidence rate of pneumococcal pneumonia was 10 per 200, right? And then we multiply by 100 to get a percentage. That's 5%. The prevalence here was 23 over 200, which gives us 11.5%. So that's a little worrying, right? 11% of your nursing home has pneumococcal pneumonia. I'd say that's an outbreak of some sort occurring, right? It's probably being transmitted from one person to another. So an outbreak is going to be marked by both an increase in incidence and an increase in prevalence. If we took steps to control this outbreak, like maybe we made everybody in the facility wear a mask if this is transmitted respiratorily, we would look next month and see, are the number of new cases going down? Are the number of active cases going down? So that would be a direct quantitative way to try and measure this. Of course, you have to use statistics and things like this, and it gets complicated quite quickly, but we have to have numerical ways of measuring this stuff. I also mentioned that graphing or representing data is critical to epidemiology. Here we can see two figures. We have the United States, and in this case, we're looking at Lyme disease incidence over time. So in 2001, any cases that occurred, we color them light green. As the years go on, we look at darker green. So we can see that in 2001, the majority of the cases were localized to the East Coast, the Midwest here, and we do have a smattering of other places in high population areas. But we then look at the subsequent years, we can see that it has started to spread eastward out from these areas. And so many of the cases that are occurring in other regions are happening later. So we can see that the disease is spreading outward from these locations. We could also look in a particular year at incidence per state of something, in this case, syphilis. So light green is low incidence per 100,000, right? So 0 to 3.8 per 100,000. Idaho, very good in 2017, low incidence of syphilis, nice job. Other states here all had low incidences. Whereas we look at southern states, we tend to see larger incidence of syphilis, or something like New York had a large incidence of it. Now, what's the cause, right? There are many hypotheses we could formulate. It could be that syphilis just can be more common in areas with large cities, right? Idaho does not have any large cities compared to California or New York or the South, things like this. So maybe those are driving the incidence numbers up. Or maybe it's social practices, or maybe it's climactic, right? We tend to see higher incidence in the southern parts of the United States versus the northern parts of the United States. Maybe climate is a factor in that. There's all kinds of different hypotheses we can generate. And this is what epidemiologists do. I'll leave you to develop your own hypothesis. Some of them maybe more biased or inflammatory than others. But yeah. Also, we can use graphs. Everybody loves graphs, right? Graphs can be boring, but graphs can be really important in telling us a lot of data in a very clear way. So often when we look at like incidence, we're measuring over time. So a graph is great for that. It doesn't necessarily give us that geographic information, but it gives us overall over time so we can look at what we did. So here we're looking at cases per 100,000, and this is for measles. So as we know, measles has dropped significantly through vaccination campaigns, but we did notice a low level until about 1990. We noticed a spike in measles. This was ultimately identified to be caused by the way we were vaccinating. So we were doing a single dose vaccination, which didn't build as strong of a long-term memory in our immune system as a 2-dose system. So after this, they switched to a 2-dose vaccination strategy, and we've seen that the numbers have dropped dramatically until recently. And these upticks in incidence Usually we'll have an uptick and lots of parents have their kids get measles and everyone hears about it and they go, most people go, I don't want my kids to get measles. I guess I'll go get them vaccinated. And then we have a drop off in it. Hopefully that doesn't continue to cycle. Hopefully we can convince people to vaccinate their children against measles before they have to get the disease. So measles used to be endemic or considered endemic in the United States. It is not considered endemic right now, although with increased incidence, that could change. Let's look at another disease, mumps. Interestingly, again, vaccination lowered the numbers. Measles, mumps, rubella, all one MMR vaccine. We saw in 2006 an outbreak, but this was in college students. So this led us to realize that actually after a certain number of years, 15, 16, 17 years after vaccination, the immunity started to wane from that single dose vaccination. So again, we could recommend like giving a booster to people after 15 years, particularly when they go off and mingle in new locations, like when students go off to college. That's a new interacting of people from many geographical areas. So after that, we identified that and we had a decrease in that. We particularly boosted healthcare workers. People were getting sick with mumps and then were coming into hospitals, and then hospital workers were getting sick with mumps because their immunity had waned as well. So in this case, we boosted the hospital workers. That helped drop it down. Finally, we have something like pertussis, which is whooping cough. Now this shows us that there are some considerations that we have to take when we look at data. So data has to be collected. If we look at this, we see that over the years, incidence of pertussis or whooping cough have actually gone up in the United States. Is that because we actually have more cases of whooping cough? Or is it because we are better at screening people and collecting data on who actually has whooping cough? Maybe in the 1980s, somebody had a whooping cough, they came in, and we just went, they just have a cold, and sent them away, and never collected that data. Maybe the internet era has allowed small clinics to better report incidences of whooping cough, right? You think about 1980 in Idaho Falls. I'm going to guess that the population was very small, right? We're talking under probably like 30,000 people. You go out to these outlying communities around us, and before the internet, they saw a case of whooping cough. And do you think they always, 100% of the time, reported that to the CDC? Or do you think maybe some people got a little lazy, right? We'd like to think that people reported it 100%, but that's not always the case. So we have to factor in different things. This is something that we're questioning about autism. We see autism rates going up. Is that because autism is actually more common, or are we better at identifying cases of autism instead of just saying, oh, they're a little bit different? We go, oh, they are different. They probably have autism. We will report that. So these are questions that can't always be answered, but something to think about. How do we get this data? Well, here's where we use what we call the tools of epidemiology. Obviously, clinical microbiology labs are super important because to report incidence of disease, you got to identify the microbe. There are often centralized labs that things can be sent to for complex sample processing. But then also, we also report that data all back to a centralized organization, the CDC. So we have different CDC labs. If we have something that is very rare, we often send them to there and they do the microbiological work and figure it out. But even if a small lab around here identifies something, we all report back to the CDC. And this is part of what we call surveillance. Surveillance has 3 stages, and often this involves databases, and computers make databases so much easier to work with. We're looking at several different things. First off, does the patient's disease fit into an already established CDC definition for a specific infection? So whooping cough, right? Someone comes in and we hear that whoop and we test it. So does their disease fit into what we know as whooping cough? Yes or no? We then try to identify the case's geographical location and get any information on the patient's demography, info on age, sex, race, income, things like this, where they go to school or things like that. We then take that information, put it all together, and identify trends. If we found that whatever's coming in doesn't fit into one of these, this data is super critical in identifying what might be the cause and how it is being transmitted. So it's important that we have both of those things, right? Is this new? Is this old? And who are we seeing this in? So that's like just collecting information on signs, symptoms, things like that. We also have what we call molecular surveillance, which is screening tests like PCR to identify non-symptomatic cases or early cases, things like this. This helps us identify things early, get treatments to patients before they interact with other people, and try to identify and stop the spread of disease before we start seeing the signs and symptoms, which is the best case. We could also use molecular surveillance to do things like test mosquitoes for the presence of Zika virus, or around here, West Nile virus, things like that. So molecular surveillance, super important, and we can get much more information beyond what we know from signs and symptoms and things like that. All right, that was a lot of information, but I think when you put it all together, it really makes sense, right? You got to collect data, go out there, find it. That's descriptive epidemiology. Then you take that data and you develop hypotheses, you analyze it. That's the analytical epidemiology, and then you test those hypotheses. We described endemic diseases, epidemics, and pandemics, dealing with increase in case number and spread in geographical area. Talked about incidence and prevalence, and some surveillance techniques here. All right, that's it for 26.1.